So let's turn to a different target, MET, which has been one that we've had some hope for, some encouraging points and some missteps. One of the more encouraging presentations was of crizotinib, or Xalkori, which Nasser, as you alluded to, is a very potent MET inhibitor, was actually kind of accidentally found to be an ALK inhibitor and diverted in that direction, which was to the great benefit of many people with an ALK rearrangement, but now is being studied full-fledged for the minority of patients who have MET amplification. Can you tell us about Dr. Camage's presentation?
Sure. Dr. Camage looked at patients with varying degrees of MET amplification. The study was rather small, but still pretty convincing. There were 16 patients, 3 had to be excluded, and then the remaining 13 were divided into groups based on how much MET was measured by FISH, which is a measurement of the gene copy number of MET. So the 3 groups were low MET amplification, intermediate MET amplification, and high MET amplification. And then he looked to see who responded. And there were 4 partial responses, 1 in a patient who had intermediate MET expression, 3 in patients with high MET amplification, I mean to say, suggesting that in this group of patients with higher copy number, those are the patients that will in fact benefit from this type of target inhibition. In this group of patients, crizotinib was well tolerated with the side effects that we have come to expect with crizotinib. And so this is an innovative study. I thought it's reusing a drug in a new way, and I think we are all trying to look for opportunities to do that with drugs that are currently available. And I think there is more to this story that will unfold in the next year.
So, any sense of what proportion of people with lung cancer could benefit from this approach?
Well, that's a good question, Jack.
It's about 1 to 2%.
Yeah.
It's not a large number. It's fewer than ALK.
I guess Dr. Capuzzo made the point that we should look for MET amplification in a different group of patients than we have typically looked in the past.
Because you see it in smokers, and you see it in squamous cell cancer, not the demographics of the ALK. But I thought, the take-home for me, there were a couple of take-homes. So this was part of a large phase 1 program, and the company did things right. They knew the targets of this drug, and so they decided to select patients who had those targets. And they've reported them independently, and of course the first report was with the ALK positive group, and Dr. Camage reported that as well. And now Dr. Camage is reporting. This is all from the same study, but now this is the patients who have MET amplification. And so it would be instructive to use that model, I think, for other drugs. There are drugs that hit PDGFR, there are drugs that hit FGFR, and the numbers are not large, but if 1 or 2% of patients have abnormalities in those genes, perhaps we can demonstrate in a 15 to 20 patient subset that you're going to have activity. So I think that that's instructive. The other thing that's instructive is that it's not sufficient to identify the gene. You really have to understand the functionality of the gene. So there are such great examples here. So with drugs like Tarceva, it doesn't matter so much if you have a lot of protein expression on the tumor cell. It probably doesn't matter so much if there are a whole bunch of different copies of the gene. What really matters is if that gene is mutated. With MET, it really doesn't seem to be that it's the expression. It doesn't seem to matter if it's mutated. What really matters, or it seems to matter, is how many copies of that gene. It's fascinating that a tumor, what makes something go from normal to abnormal isn't necessarily because that gene is altered. It's just that there's too much of that gene. Instead of there being the normal number of copies, for reasons that we just don't understand, your cell aberrantly produces lots and lots of copies. And so too much of a good thing is a bad thing, in this case with cancer. So it's not enough just to understand. And with ALK, it's completely different. With ALK, it's not expression. With ALK, it has nothing to do with a mutation, although ALK is mutated in some tumors, interestingly. It doesn't matter if you have a whole bunch of copies of ALK. Crizotinib is not active if you have just a bunch of copies. What makes it functional is if there's a rearrangement where one gene should not be next to another gene, and for reasons we don't understand, and this happens a lot in cancer, that gene is rearranged next to another gene and it activates that gene in an aberrant fashion. So we need to understand not only what genes are in play, but why they're functionally causing cancer.
Would crizotinib be a potentially effective approach for MET mechanisms in acquired resistance to EGFR?
We don't know. And it's purported that that is a mechanism in 10 to 20%. It actually is probably a little bit less than what we originally heard. But Melissa, have you seen data on crizotinib in patients who've progressed on EGFR TKIs who now have MET amplification as a mechanism of resistance? I'm not aware of it.
It's just a thought.
I mean, we can talk in a minute about a different way of inhibiting MET that didn't work, that may not work so well for patients with acquired resistance. But this is a good question that probably could be investigated.
Yeah.